aims and objectives of this topic are to make students know what a fossil is and the domain of fossil definition, to make them understand how fossilization occurs, to throw light on various types of fossils, and to highlight fossil importance. The word fossil literally means something dug up, but it is now used in the technical sense for the remains or traces of animals and plants which lived under conditions different from those of the present. The science of burial is called taxonomy. Fossils act as an evidence of past life. Fossilization is the process of fossil formation. Fossilization can preserve actual remains of an organism or evidence of their presence in an ecosystem. Very small percentage of organisms are immortalized in stone, that is fossilized. Fossilization takes a lot of time. It needs appropriate climate and topological conditions. All organisms and all the parts of an organism are not fossilized equally. Hard parts like bones and xylem are good for fossilization. There are different types of fossils such as casts, molds, petrifications, and impressions. Fossils are found in sedimentary rocks and are formed by different types of fossilization. Fossils are proving important in diverse ways ranging from evolutionary to commercial uses. Fossils reveal the structure of extinct organisms. They also help in solving the puzzle of origin and evolution of different organisms. Fossils are the mineralized or otherwise preserved remains or traces of animals, plants, and other organisms. The totality of fossils and their placement in fossiliferous rocks, that is fossil-containing rocks, and sedimentary layers, that is strata, is known as the fossil record. The earliest fossils are about 3,100 billion years old. They are of simple prokaryotic organisms like bacteria and cyanobacteria. Earlier fossils were considered products of stars and thunderbolt, but Leonardo da Vinci and Bernard Palissy were the first to report that they are the past organisms which lived on the earth. 400 million years old fossil plants are the oldest known examples of wood. Now, which thing should be regarded as fossil? Fossil definition needs careful consideration. The term fossil is used for any trace of past life. Fossils are not only the actual remains of organisms such as teeth, bones, shell, and leaves, that is bodily fossils, but also the result of their activity such as burrows and footprints, that is trace fossils, and organic compounds they produce by biochemical processes, that is chemical fossils. The remains of plants as in coal or lignite are usually found in the form of carbon. The remains of animals usually consist of hard parts of bones, teeth, or shells. Traces are such things as footprints, coprolite, that is fossil dung, or faces, dwellings, burrows, and eggs. Conditions different from those of the present normally excludes an organism which have lived in the past 1,000 years or so. The mummies of an ancient Egypt or Inca or the skeleton of a Bronze Age donkey would not be regarded as a fossil. Another definition of fossil is that they are the remains of animals and plants which lived prior to historical times, that is prehistory. Their study is a specialized branch of geology known as paleontology. Scope defined fossil as any specimen that demonstrated the physical evidence of the occurrence of ancient life, that is Holocene or older, may be regarded as fossils. Sometimes a demarcation is made between a fossil and a pseudo-fossil. Fossil is regarded as an older specimen which has been fossilized before the end of the Pleistocene period. They are completely mineralized and thus changed into fossil. Subfossil is a recent dead body of an organism less than 6,000 years old and is less mineralized. Steward's definition of fossil as any evidence of prehistoric life seems to be an exclusive definition. Some fossil-related terms are also common, namely pseudofossils, living fossils, and index fossils. Index fossils. These fossils are used in dating of rocks or act as indicators of petroleum and coal. They are usually past organisms which were small in size and lived abundantly with unique physical characteristics. Now, living fossils. It is an organism which shows long geographical record and is still living in a limited area. It is not a fossil, but it is the oldest representative of a fossil group of organisms which dominated the earth in the past. Living fossil is a term used for any living species that closely resembles a species known from the fossil. It is as if the fossil had come to life. Best example is furnished by Ginkgo biloba. The genus formed forests of Jurassic period, then became extinct and is now represented by few representatives only. Now, pseudofossil. These are actually non-living things like rocks which give an illusion of fossil of plant or animal. Now, where to hunt for the fossils? Laserstratum are the world's best fossil sites. Fossils are found in rocks. Fossils are found in sedimentary rocks, mainly limestone, siltstone, and mudstone that were deposited in former lakes, rivers, estuaries, seas, and caves. Sedimentary rocks are formed as a result of deposition of various kinds of sediments like gravel, sand, clay upon the land surface or in the water bodies. These sediments are finally converted into rocks after a long interval of time. Since these rocks are formed in layers, living organisms may become fossilized in them and are available to be dug out by chance. Some of the common sedimentary rocks are the limestone, gypsum, shale, diatomite, rock salt, and coal. How are fossils formed? Organisms pass through a number of stages on their way to become fossils. The important stages are death. Generally, this is the first stage in fossilization. Death can occur as a result of burial, and in this case, biostratinomic processes may not take place. Biostratinomic processes include reorientation, disarticulation, fragmentation, and corrosion, which modify the remains. Thus, information is lost. Another stage is deposition. Burial of the remains in sediment occurs where they are less likely to be disturbed. Plant parts are best preserved in the very fine-grained silicates and shales, which are the lithified muds of the ancient deposits. Such sediments generally yield excellent fossils because the small grain size preserves minute details of the leaf. Coarser-grained sediments such as sands generally do not reproduce delicate features. Plant parts composed of thick-walled cells or those having fibers within their tissues have a better chance of being preserved, that is, good fossilization potential. Now, diagenetic processes. Diagenetic processes include chemical or physical changes, that is, permeation and carbonization. These are necessary for fossilization. Now, optimum conditions for preservation. There are two major requirements for an organism to be preserved as a fossil. Number one, it must possess hard parts. Number two, it must undergo rapid burial in a productive medium. Various agents can be responsible for the preservation of organisms. Commonly, sediments bury the remains and keep them from being disturbed during the process of fossilization. More unusual agents include lava or volcanic ash and severe cold or aridity. Firstly, the organism must be deposited in sediment. Between the time of death and the burial, biostratinomic processes alter the remains. Before burial, an organic skeleton is normally subjected to reorientation, disarticulation, fragmentation, and corrosion. Once it is buried, it undergoes diagenetic processes. Such changes are necessary for preservation because organic matter will not survive for long before it is decomposed, and hard parts as bones, teeth, calcified shells, xylem, sclerenchyma are normally prone to destruction. Now, carbonization. Carbonization is a process occurring within the sediment in the absence of oxygen. In such conditions, anaerobic bacteria consume hydrogen and oxygen, therefore concentrating carbon. The final products of carbonization are coal and hydrocarbons, which can be considered as accumulations of very badly preserved fossils. Now, permineralization. Permineralization occurs when the plant tissues are infiltrated with mineral-rich fluid. Minerals, commonly silica, carbonate, phosphate, or pyrite, precipitate in cell lumen and intercellular spaces, thus preserving internal structure of plant parts in 3 dimensions. When mineral matter actually replaces the cell wall and other internal structures, the preservation may be called petrification. Following are important types of permineralization. Silica permineralization. Silification commonly occurs in rare areas where silica-rich volcanoclastic segments are weathering. For example, the famous upright trees in Yellowstone National Park. According to Leo et al., concentrated watery solution of minerals like salic acid infiltrates the tissues, which is followed by intracellular or intercellular polymerization of siloxane bonds and elimination of water. With the continuous growth of the polymer, the amorphous silica gets deposited from the solution and the water moves out of the plant tissue. Ultimately, this silica gets converted into quartz and fossil gets petrified. Silica, and especially its most common crystallized form, the mineral quartz, is extremely stable at the Earth's surface. Therefore, silified fossils are likely to withstand modification or damage. Permineralization with calcium carbonate, calcite, or dolomite is particularly common in Carboniferous coal seams where whole regions of peat were permineralized. Called as coal balls because of their round or ellipsoidal shape. Now, carbonate preservation. Ideal conditions for carbonate preservation are normally found in marine and shell sediments with a high biomass of organisms that could potentially fossilize. Marine waters are constantly supersaturated with respect to calcium carbonate, and the preservation of carbonate shells is rather common instance. Now, pyritization. Pyrites are iron sulfur minerals. These pyritized fossils often occur in the presence of seawater, which is the source of sulfur, and are characteristic of plant tissues washed into marine basins. Permineralization with phosphate. Phosphatization of soft plants have an unsurpassed degree of preservation with detailed cellular structures. Muscle fibers are most often preserved through phosphate replacement. Now, charcoalification. Charcoalification can preserve tissues burned in wildfires or buried in pyroclastic flows. Internal structure is often detailed. There are 6 main types of fossils based on the manner of fossilization. These are coalified compression. It is a fossil containing compressed coalified remains of plants. Another one is authogenic preservation or cementation. Here, surface layer of the organism or part is preserved by cementation. In cementation, compact sediments are cemented by cemented material like silica or calcium to form sedimentary rock. It shows only the surface detail of the fossilized organism. Following types are its examples. Impressions, also called imprints, are the external features of the past organism or their parts left in the hard rock. Another one is mold. These are encasements of the hardened mud or minerals that surround a fossilized organism. Another one is cast. They are hardened minerals deposited inside the mold. Infiltrated cellular preservations. Here, cells and tissues of the plant or plant parts are preserved. It shows internal detail of the organism. It includes following types: petrifications, also called true fossils, because in them cellular details are preserved due to replacement of organic matter particle by mineral matter. Another one is mummification. In this type, the entire organism or body part is preserved in some special preserving material so that all its soft body parts are preserved perfectly and remain intact. Thus, organic matter has not been replaced by mineral matter in such fossils. For example, ice preserved fossils. Another type is hard part preservations. Here the plant possessing hard parts are preserved, for example diatoms. Now structural fusionites. The qualified plants after charring in forest fire form fossil charcoal or fusan are called structural fusionites. These fossils show cellular detail, though cell walls are generally absent. Now the chemical fossils. These include fossilized plant chemicals like chlorophyll, DNA and proteins. Now let me explain these fossil types in detail. Qualified compression. It is a type of fossil that contains compressed qualified remains of plants, generally in the form of black carbonaceous film in the rock. The compression retains the original outline of the organism. Compressions are plant parts that have suffered physical deformation such that the three-dimensional plant part is compressed to more or less two-dimensional. Compression mainly occurs in plants retaining qualified organic matter. Well-fossilized compressions of leaves show leaf form, veins, stomata, epidermal hairs, and cuticle. Coal is the best example of compressions. Now fossilization of compressions. Plant or plant parts get buried in sediment layers and under high pressure gets converted into coal. Generally, compressions retain their lower surface intact while as upper surface gets distorted. Plant stems and other three-dimensional plant structures do not preserve well under compressions. In carbonation, the decomposition of organic matter under anaerobic conditions or water, the hydrogen, oxygen and nitrogen are driven off, leaving the carbon residue. This leaves a thin layer of carbon on the rock as evidence of the original organism. Now, what are impressions? Impressions are two-dimensional imprints of an organism that do not contain any organic material. Thus, impressions are essentially compressions without any organic material. Impressions probably represent the most common type of plant fossils. They are formed by dorsiventral or two-dimensional plant structures like leaves and sporophylls. They occur when a plant part is covered by sediment and the water is squeezed from the cells and tissues. Cells that make up the plant are generally degraded, perhaps as a result of microbial activity, so that only a shallow negative or imprint of the plant organ, that is leaf, remains. Impressions usually occur in the fine-grained sediments such as clay or silt. Impressions may also occur if, when the layers of rock are split apart, the organic material adheres to only one side of the rock. In this case, the side with the organic material is the compression, known as the part, while the corresponding impression known as the counterpart. Now, let us see what do impressions tell us. Paleobotanists determine the shape, venation and other structural features of the leaf and notes any characteristics that make it unusual. An impression is a clue left of the organism's biological activity. Some examples of impressions are the trace fossils such as footprints, traces, remains of tunnels left by the burrowing organism, and the holes made in the shell by parasites. When sediment is deposited into the cavities left by the decay of the plant parts, a cast results. A mold is essentially a cavity left in the sediment by the decayed plant tissue. Molds are generally unfilled or may be partially filled with the sediment. In other words, after decay of the organism, when all that is left in an organism-shaped hole in the rock, it is called a mold fossil or triphyllite. If this hole subsequently has been filled with other minerals, it is called a cast fossil and is considered a replacement fossil. Since the original material has been completely replaced by new unrelated ones. Casts and molds may be found together with the cast filling the mold. Molds are formed when the soft sediment surrounding the structure lithifies or hardens before the structure decays. When the mold fills in with sediment that subsequently hardens, a cast is formed. Cast of an internal hollow structure like a pith cavity are also common. Like compressions and impressions, casts and molds record external, sometimes internal organ features well. Unlike compressions or impressions, molds and casts often are truer records of the original three-dimensional shape of the structure. Fossilization of impressions, casts and molds. After burial of the organism in fine clay, two processes occur, namely decay of the internal organic material of the buried plant and cementation of the surface sediment. Decaying material develops electric charge which attracts colloidal particles having opposite charge from the surrounding medium. Decaying material creates a specific geochemical situation around themselves. Sedimentary material containing iron and carbonate surround the organism and become cemented. This cemented layer preserves the surface features of the organism. Cast is formed by infilling of mold with mineral matter. Now, petrifications. Petrification means turning into stone. In these, fossil tissues are fully or partially replaced by mineral matter so that even cellular details are preserved. These are regarded as the best fossils for the study of internal organization of the fossilized plant. Now, fossilization process of petrification. After burial of the plant in sediment, penetration of silicates, iron compounds, and carbonates starts between and inside the cells. According to Leo et al., concentrated watery solution of minerals like salic acid infiltrates the tissues, which is followed by intracellular or intercellular polymorphism of silicon bonds and the elimination of water. With the continuous growth of the polymer, the compound silica gets deposited from the solution and water moves out of the plant tissues. Ultimately, this silica gets converted into quartz and fossil gets petrified. These represent the actual remains of an organism which remain fossilized in ice, volcanic ash, or old bogs. There are following types. Ice-embedded fossils. Fossils of mammoths and rhinoceros have been found in the Siberian ice. They belong to late Pleistocene. Their flesh, skin, and hair was intact. Preservation in oil-saturated environment. Organisms fossilized in oil-saturated places preserve their organic parts. Tree trunks found in such places are so well preserved that it is possible to cut them with saw and drive nails. Another one is resin-embedded fossils. Amber is a fossilized resin which can preserve insects and pollen grains. Fossilized pollen or spores are called microfossils or palinofossils. Fossilized insects of the Oligocene period have been found preserved in the amber. Thus, amber acts as a nature's tomb. Amber forms when plant resin is exposed to the air, causing its constituent molecules, that is terpenes, to cross-link and harden. The substance is very resistant to decay, with the oldest amber found dating to late Carboniferous period. Amber younger than 40,000 years is termed as subfossil. This is highly useful if the amber has plants or animals trapped inside. Organisms are three-dimensional with excellent morphological preservation, but their internal structure is dissolved. Now, bog people. Peat bogs have yielded the famous bog people, 2,000 years old humans with intact skin, hair, and clothing due to the presence of antimicrobial, tannic, and fulvic acids. Because of the low pH, their bones have been dissolved. Now, hard part preservation. Some resistant plant materials like silica and calcium compounds are immune to physiochemical changes, thus serving in fossilization of the organism. For example, diatoms containing siliceous fascicules are the best fossilized plants. Another one is structural fusionite. The coalified plants, after charring in the forest fire, form fossil charcoal or fusion are called structural fusionites. These fossils show similar details, though cell walls are generally absent. These are also called chemical fossils. As more become known about the chemistry of modern plants, paleobotanists have begun to examine the fossil records for corresponding chemical data. For example, characteristic breakdown of chlorophylls and lignins have been found in well-preserved fossil leaves. Lipids and their derivatives have also been recorded from sediments. Some carbohydrate breakdown products may also survive in sediment. A special class of these oleoins are formed by the flowering plants, some ferns and lichens. An increase in abundance of these molecules in sediments of mid to late Cretaceous is used to document the increasing abundance of flowering plants. In another study, genetic material was recovered from tertiary leaves.